Hi everybody, so today we're going to be talking about the role that NOD2 mutations play in Crohn's disease. And before we get started, what I'm going to do is I'm going to go over what NOD2 does under normal conditions. So what we're looking at here is the digestive tract right here. So if we look up here, this is going to be the esophagus. The esophagus carries food from the throat down to the stomach. And you're looking at someone this way, okay, as if they're facing you. This here is going to be the stomach. Then what's going to happen is we're going to have the small intestine. All this red part in here is the small intestine, okay? So this part's called the ileum, and we're gonna come back to the ileum in just a minute. This green part is going to be the large intestine. So food's gonna go from the esophagus to the stomach, into the small intestine, into the large intestine, then out of the body. The part that we're most concerned about is when someone has a NOD2 mutation in Crohn's disease, is right down here, we call this the terminal ileum. To give you an idea of where this is at, this would be where your appendix is at. So looking at here, it'd be way down here on the right-hand side. Okay, so that's where we're basically going to see the symptoms from a NOD2 mutation. Now, can they occur in other places of the digestive tract? Yes. But let's start right here. So what I'm going to do is I'm going to take a cross-section. Before I get there, imagine it's like a hose as food is broken down and it's going back and forth and it's going through this tube here. So what I'm going to do is I'm going to take this portion of this and I'm going to pull it out, and I'm going to go like this. And like I said, it's a tube. Imagine it like a hose. This would be where the water is. And then I'm going to put it up on here. If we look here, we have all these tissues here, right? Which I don't have all the layers drawn. I'm going to go over those in just a minute. And then we have the opening on the inside, which we are going to call the lumen. There's the lumen. So again, if this was a hose, this would be where the water is. This would be the rubber part around it. What I'm going to do now is I'm going to take a portion of this and I'm going to take a portion, I'm going to pull it out, I'm going to stick it right here, and now that's what we're looking at right there. So, this here, we have this layers here, right? What I said would be the rubber on the hose, that's these layers here. So from here up to here is going to be my mucosa. Then under that, we're going to have the submucosa. By the way, this little piece of brown, that's just a thin muscular layer right there. Then we're going to have the submucosa, we're going to have the muscular layer, and then we're going to have the serosa. And we're going to come back to these in a little bit. If we look here, see the red that we have right there? These are epithelial cells, and they're basically going to line the digestive tract. Okay, so now, if you notice these here, and I only have two drawn, but if you notice these here, and you have these all over the intestines, these are going to be called goblet cells. And what goblet cells do is they're going to secrete a fluid that's kind of like saliva, and it's called mucin. So we're going to get mucin coming out from here. And it's going to line the digestive tract. Okay, it's just going to line the digestive tract. And what this mucin is going to do is it contains proteins and it helps keep bacteria from entering into these layers that are here. So it's going to keep them out there. The other thing is if you notice right here, we have these purple cells. This part that comes down here, these are called the crypts of Lieberkühn. And what happens is at the bottom of these we have these little cells, and we're going to call these Paneth cells. Okay, so we call these Paneth cells. And what these do is these are going to secrete a substance called defensins that's going to help fight off microbes. So if we look real quick, let's say we're getting some microbes in here, right? So we got some microbes in there, and we don't want those there. This is going to secrete defensins. And so what I'm going to do now is I am going to take one of these microbes, and I'm going to blow it up and make it bigger, right? So here's my bad guy, right? There's my bacteria. And then these have a cell wall on them. So here's my cell wall on this. And the cell wall is going to be important in a little bit. That's why I'm drawing it. And what's going to happen now is the defensins is basically going to come over to this microbe, and it's going to basically poke holes in it. It's going to basically stab it and kill it. And that's the way defensins work. So NOD2 plays a role in these Paneth cells making defensins. So that's two ways that NOD2 plays a role in the immune system in your intestines. The third one's going to be is if you notice these cells are really tight together, and we call these tight junctions. And the reason you have tight junctions is because these are so close together what happens is they keep microbes from coming in also. So we have three ways that NOD2 is already going to protect your intestines. And basically what it is you have the tight junctions. Like I said, they're really, really tight together. They're slammed together, right? So that keeps microbes from squeezing through there. We have the pannate cells releasing defensins, which is going to basically stab the microbes that are there. And then we have the goblet cells secreting mucin. All right, so here's my mucin right here. You have the goblet cells secreting mucin. So that's three ways that NOD2 is going to protect us. Now let's look at something else that NOD2 does, and we'll talk about the role of the mutation in this. So if we go right about here, what I'm going to do is I'm going to take one of these epithelial cells here, okay? Now I'm going to take an epithelial cell, but this could be a macrophage, which is a type of white blood cell. Also, it can be a dendritic cell, okay? But I'm just going to use this here, and I'm going to make this larger. So okay, and so here's my epithelial cell. And now, here's what's going to happen. Inside the epithelial cell, like we said, we have something called NOD2. Now, NOD2 is kind of shaped more like a Y, but this is going to be my NOD2. Okay? When I get this microbe here, that's here, what's going to happen is it's going to lose a piece of its cell wall. Piece of its cell wall is going to come off. And we call these pieces, they're going to be called monomer dipeptides. And then what's going to happen is these are going to enter into the cell, and when they do, they're going to activate this NOD2. Now on the inside of your cell, you also have something called DNA. So here's my DNA, or here's my nucleus, and then inside the nucleus, I have DNA. So when this NOD2 becomes activated, I'm not going to go through all the steps, but what's going to happen when this NOD2 becomes activated, it's going to cause the activation of something called nuclear factor kappa B. Okay, nuclear factor kappa B. Then what's going to happen is the nuclear factor kappa B is going to basically, it's a transcriptase, so it's going to latch onto that DNA and it's going to cause the release of cytokines. Cytokines are basically substances that are going to cause inflammation. The cytokine it's going to make is interleukin 1, interleukin 6, interleukin 8, and then something called tumor necrotic factor alpha. That's what it's going to make under normal conditions. Okay, that's what we want, right? Because we're protecting our immune system here. The other thing that's going to happen with this NOD2 is it is going to go and it is going to cause something else in here called ATG16L1. And this is going to be basically another protein. Let's draw this guy below. So this is going to be another protein that's inside of our cell. And then what this is going to do is it has two roles. The first thing it's going to do, well, it's responsible for something we call autophagy. Autophagy is basically when the cell eats itself, right? It's going to break itself down when it gets old, so it can be replaced by a new epithelial cell. What's going to happen here, though, is the other role this is going to play is, you see how we have this microbe here? Now let's pretend for a second this is a macrophage, or it's a dendritic cell. What it will do is it will actually grow the cell membrane out, so it will surround this here. These are called pseudopods. It will surround this here, right, with these pseudopods, and then it's going to pull it inside the body. So if we go like this, if you notice, I still have my cell membrane here. So it's going to pull this inside the body, and I'm just going to draw this down here. All right, and now I have it right there. I'm not going to put the cell wall in there. But also inside of here, oh, and by the way, when this pulls this inside, this is going to be called a phagosome. because it did something called phagocytosis. When you go and you grab those and you pull them inside, that's called phagocytosis. And now what's going to happen is we have another type of vesicle in here, which we're going to call a lysosome. And lysosomes have something inside of them called hydrolytic enzymes, right? And that's going to be these here. All right? And then what's going to happen is this lysosome is basically going to form a connection with this phagosome. And when it does that, now these hydrolytic enzymes are going to come and they're going to break this up. They're going to kill this guy. Okay, they're going to break it up. And that's what it's supposed to do, right? That's under normal conditions. The other thing this ATG16L1 is going to do is it's going to come up when we don't need this anymore. So let's say we've cleared out all the bacteria there and we don't need it anymore. It's going to come over here and it's going to inhibit the nuclear factor kappa B from making more of these. And then we stop making those. Okay? Everything so far is under normal conditions, right? That's what's going to happen. So now we're going to work our way backwards and look what's happened with Crohn's disease. So in Crohn's disease, first of all, in Crohn's disease, it's considered an autoimmune disorder, but actually it's a dysregulation of the immune system. We get a hyper response of the immune system to microbes. So what's going to happen here? We have our mutation now, right, of this NOD2. When you get the mutation of NOD2, and these here are called, those myramyl dipeptides is called MDP, okay? So now, when you have a NOD2 mutation, it doesn't recognize the MDP anymore. So these come in, but the NOD2 just doesn't know they're there. When that occurs now, we're not gonna get this nuclear factor KB activated, which means we're not gonna get any of these, right? So now I'm not gonna get my immune response. What that means now is I am gonna start to get microbes building up in this area. Okay, so that's the first thing that's gonna happen in the NOD2. I'm not gonna get my immune response, okay? The second thing that's gonna happen is, you see this ATG16L1, that's not gonna happen. So where it's going around and it's pulling this in and it's destroying it, this is no longer gonna happen also. So now I'm not killing these. So again, I'm gonna start to get my microbes building up here in my digestive tract, right? The third thing that's gonna happen, it doesn't really matter right now, but we'll see in a little bit, we don't turn this off. But here's what's gonna happen, is you have this ATG16L1, you also have an area on this that we're gonna call T300, T300A. You can get a mutation in this also. If I get a mutation in this, what's gonna happen is my MDP will come, it will activate the NOD2. The NOD2 will still activate this, but now we have this mutation, so none of this happens, right? And once again, because of the fact that we have this mutation here, we're not gonna get this. We're not gonna get the phagosomes pulling this in, the pseudopods pulling this in, we're not gonna get the lysosome binding to this, and we're not gonna be killing bacteria. The other thing that's gonna happen in this case is this is not gonna shut this off, right? So now we're producing too much of this. This is not gonna stop when it's supposed to stop. We just continue to produce too much of it. Okay, so there's a few things that can happen. Just to recap real quick, I can get a NOD2 mutation which doesn't recognize the NDP, so I don't make any of this, which allows microbes to grow. Also, if I have the NOD2 mutation and it doesn't recognize this is not going to occur, so it doesn't do the phagocytosis, and therefore it doesn't kill the bacteria, which allows the bacteria to grow. The other thing that can happen is I can get a mutation on the ATG16L1 at the T300 area, and then what's gonna happen is my NOD2 still works, but this doesn't, so it doesn't inhibit this when it's supposed to. So now I'm gonna get a hyperimmune response. So that's a lot. So now let's take a look at this. This is what's also gonna happen. If I have the NOD2 mutation and none of this is occurring now, we have to find an alternate pathway, an alternate inflammatory pathway to basically make this here, to fight off these infections here. So what's going to happen now is on your cells, okay, and this would actually be on the part that's exposed, not on the inside in here. But what's going to happen now is you're going to have something that we call a toll-like receptor. And these bacteria, their cell walls also secrete something that we call PAMP. Pathogen-associated molecular pattern. So what this is going to do is it's going to be a piece of this cell wall on a bacteria will come off and it will bind to this toll-like receptor. When it does that, what this is going to do is it's going to make 3 different types of cytokines. Remember, cytokines produce inflammation. So the first one is going to produce something called NLRP3 inflammosome. It's also going to produce interleukin-1, which you saw it was right here. It's also going to produce something called interleukin-18. So we're going to come back to this in just a minute, but what I want to do is I want to talk some more about what these NOD2 mutations do. So if we have a mutation in NOD2, if you recall over here, what we said is NOD2 is responsible for making this mucin. So when you have a NOD2 mutation, you do not make this mucin anymore. And it's going to stop. So now what's going to happen is that's the first line of defense against pathogens getting down here inside the digestive tract in the mucosa. So that's gone with the NOD2 mutation. If you recall these tight junctions where we said they're really tight together because they're so tight, microbes cannot get in. Well, what will happen is these will start to break down, those tight junctions. And now I'm going to start to get microbes down here in this mucosa area. If you recall also, we said on these Paneth cells, we had the defensins. And so what's going to happen if you have the NOD2 mutation also, you're not going to make defensins anymore, so you're not going to kill the bacteria. So like I said, you're going to start to get all of these microbes building up in here, and your body's not fighting them off like they should. So now, if we go back over here, what's going to happen is, like I said, we're using this alternative inflammatory pathway. So what's going to happen now is normally, when this is done, by the way, with the NOD2 mutations, normally when we don't need this anymore, this NFKB will actually inhibit this. It'll shut this down when it's not needed anymore. But again, because we have a NOD2 mutation, that's not going to happen. So now this is going to be produced uncontrollably. So let's say we have a macrophage now. So here's my macrophage. And in this macrophage, it is releasing this interleukin-1. And it's also releasing this interleukin-18. And it's also going to release tumor necrotic factor alpha, like you saw was being released here when the NFKB was working. Interleukin-18 is basically going to bring T1 helper cells over to this area. And T1 helper cells are going to secrete something called intrinsic factor, and it's also going to secrete tumor necrotic factor alpha also. So I have tumor necrotic factor alpha being produced here and here. Now, like I said, these tight junctions are going to be broken, and we're going to start getting microbes coming in here. So here's my microbes now in this mucosa area. And what my white blood cells will do, and other immune cells, is they will start surrounding these microbes in here. And they do this to try to isolate them, to keep them from going to other areas. So eventually you get so many of these immune cells here, it forms a mass. I'm going to call this a granuloma. Okay? The other thing is because of the fact that there's nothing to inhibit this tumor necrotic factor from being released, as well as these other interleukins or these other cytokines, even though they may clear out the microbes, because there's nothing to inhibit them, they'll still be being released. And if we recall over here when we said we had the Paneth cells, well, they're going to destroy the Paneth cells, and they're just going to keep eating away at the lining of the intestines. So they're going to eat away through the mucosa, they're going to make their way down and eat their way through the submucosa, this muscular layer, and then into the serosa. And this is going to cause ulcers in there. So I'm going to show my inflammation like this. And you're going to get all this inflammation that's in here that has eaten away the inside of my digestive tract. So what we're going to call this now this big ulcer here, we're going to call this a transmural. We're going to call this a transmural ulcer. Transmural ulcer basically goes from the mucosa to the serosa, and it can actually even go through this and basically form a connection with something else, such as the urinary bladder. So it can and then we call that a fistula. Okay, so we're going to have uncontrolled release of cytokines. We're going to lose our tight junctions. We're going to stop making mucin. We're going to stop having defensin. I'm going to get microbes that are going to start coming into this area. We're going to form a granuloma, and these cytokines are eventually going to eat away at the inside of our intestinal wall. So that's it for Crohn's disease and the effect of NOD2. If you liked the video, please hit the like and subscribe button, and we will catch you next time.